In 1928, a single, overlooked stain in a forgotten petri dish began a chain of events that would change medicine forever. Before antibiotics, infections were a slow trap. Minor cuts could turn deadly. Pneumonia filled lungs with fluid, and childbirth fever killed mothers, because doctors had almost nothing that could reliably stop bacteria. Alexander Fleming didn't set out to cure anyone. He worked in a crowded lab, studying germs in petri dishes and recording what he saw. One morning, he noticed something wrong. A dish he had left uncovered had become contaminated by mold. Most scientists would have tossed it immediately, but Fleming looked closer because he believed the details mattered. Step by step, the story writes itself. He observed that the mold had grown in one area while the bacteria around it seemed to stop spreading. He examined the border where mold met bacteria and saw a clear zone of no growth, as if the mold had released a chemical weapon. He cultured the mold again to confirm it wasn't a fluke. The result was consistent. Where the mold lived, bacteria failed. Then came the hard part, recognition. Fleming's discovery was real, but it didn't become medicine overnight. Many researchers either didn't notice the significance, or they couldn't reproduce the effect reliably, partly because the mold was difficult to grow and the active substance was unstable. Funding and attention went elsewhere, and the world kept facing infections with the same limited tools. Fleming's idea didn't disappear. Years later, scientists learned how to isolate the substance from the mold, purify it, and, crucially, make it in large quantities. Mass production turned a lab observation into a drug that could be manufactured, distributed, and trusted. When penicillin finally entered hospitals, treatment changed from desperate waiting to targeted attack, saving lives across continents. This is how an accidental mold discovery, ignored at first, became penicillin, and why the next time you hear about antibiotics, you're really hearing about one scientist's decision to look, not just throw away what seemed broken. How mold was accidentally discovered. When European settlers stored food and medical supplies in damp conditions, they kept running into a stubborn problem. Invisible contamination that spoiled crops, grew on old bread, and sometimes ruined medicines long before anyone understood what it was. Mold was treated as a nuisance, not as evidence of a living chemical defense. Even in well-run laboratories, leftover cultures, dust in the air, and a careless streak of moisture could turn a clean experiment into a colony of fuzzy growth. Alexander Fleming's story begins the same way, small, practical, and imperfect. He worked with bacteria in a laboratory setting where petri dishes and culture plates were used to grow microbes for observation. After tending his cultures, he left them in the lab for a while, and when he returned, he noticed something that didn't match his expectations. One plate had become contaminated with mold, and around the mold growth the bacterial growth looked disturbed, as if it had been pushed back. Fleming didn't immediately announce a miracle cure. He first observed the scene carefully, comparing the contaminated area with parts of the plate where bacteria should have grown normally. The contrast was visible. Mold spread, bacteria failed to flourish nearby. He then connected the pattern to a simple question. Was the mold merely occupying space, or was it doing something chemical that harmed the bacteria? Instead of assuming coincidence, he focused on the boundary where mold and bacteria met, because that edge showed the strongest hint of cause and effect. Next came the uncomfortable part, the part that made the discovery easy to overlook. He needed to consider that the mold might be living on the plate only briefly, or that the bacteria might have died for unrelated reasons. Contamination could be random, uneven, and misleading. Laboratories at the time often produced confusing results from mixed cultures, and many scientists learned to distrust dramatic visuals unless the effect could be reproduced reliably. In that atmosphere, a contaminated plate could easily be filed away as an accident rather than a clue. Even when the antibacterial effect was real, the significance was easy to miss because the substance responsible wasn't yet usable medicine. Fleming knew the mold influenced bacterial growth, but turning that influence into a stable treatment required extracting, purifying, and testing an agent that could work outside the dish. He also faced the fact that mold products could be variable and difficult to preserve, especially when researchers lacked methods to reliably isolate the active compound. Many colleagues would have seen a fascinating observation trapped inside an uncontrolled experiment, not a practical tool for hospitals. That is why the discovery was initially ignored. The lab culture was accidental, the mold was unwanted, and the antibacterial zone could be dismissed as a one-off quirk of that particular contamination. The urgent need for antibiotics didn't mean every odd effect was automatically recognized as a life-saving path, especially when clinical medicine had long relied on supportive care and existing antiseptics rather than miracle drugs. Before antibiotics existed, treatment of infections depended on cleaning wounds, preventing spread, using antiseptics, and hoping the body would overcome the infection. Physicians had few options when bacteria multiplied unchecked in the lungs, skin, blood, or deep tissue. Eventually, the mold observation was recognized as something more than a strange plague. Researchers worked to isolate the antibacterial principle, confirm its consistent activity, and develop ways to produce it in larger quantities. That shift, from a single contaminated dish to controlled preparation, made penicillin possible beyond a laboratory curiosity. Once production could be scaled reliably, infections that used to be feared because of how quickly they worsened became treatable in a way that changed medicine at a global level. The accidental mold discovery mattered most once it was transformed into a drug that could be manufactured, shipped, and used, turning bacterial danger from an uncertain battle into a targeted, evidence-driven therapy. Why doctors ignored the discovery? Before antibiotics existed, doctors fought infections with approaches that were often blunt and risky. They relied on cleaning wounds as best they could, draining abscesses, and using supportive care while the body tried to recover. For many illnesses, pneumonia, wound infections, bloodstream infections, there were few dependable tools once bacteria had established themselves, and death could come quickly even when the cause was recognized. That makes Alexander Fleming's accidental mold observation feel almost impossible at first. He had been working with bacterial cultures in a laboratory where dishes of microbes were meant to stay clean. One day, something ordinary went wrong. Contamination appeared in a culture dish. The bacteria around it failed to thrive, as if the mold had released something that pushed the microbes back. Fleming didn't start with grand theories. He noticed the mismatch between expected growth and what actually happened, and he looked closely at the clear zone where bacteria were absent. He then followed a careful chain of reasoning step by step. First, he identified that the inhibition matched the presence of the mold, not a missing ingredient or a change in temperature. Next, he compared the affected area with regions of the same dish where the bacteria were still able to grow, which helped show that the effect was localized. After that, he worked to understand that the mold was producing an active substance rather than simply blocking the surface by physical presence. Even when he recognized that the substance had antibacterial power, he faced a hard practical barrier. Substances in a lab dish do not automatically become stable medicines you can reliably produce and administer. Doctors ignored the discovery for reasons that were mostly practical, not because they were careless. At the time, many claims about something that kills germs were unreliable, hard to reproduce, or too weak once moved from a dish to the body. A mold that clears bacteria in a petri dish could still fail in a living patient if the compound broke down, was too toxic, or did not reach the infection site in sufficient strength. Fleming's finding was also tied to a behavior that laboratories could struggle to control. The mold had to be grown and managed in a way that preserved its antibacterial effect, and results depended on conditions that could vary between experiments. Skepticism grew as researchers compared this kind of observation to past disappointments. Physicians were wary because the medical world had already seen promising antiseptic approaches that did not specifically target bacteria inside the body, and some early attempts harmed tissues more than they helped. Antibiotic-like effects were not yet a trusted category, so a discovery that required a new way of thinking had to overcome the burden of proof. Even if the mechanism sounded promising, doctors needed consistent potency, safe dosing guidance, and evidence that treatments improved outcomes rather than just stopping growth on a lab surface. Only later did the path from observation to medicine become believable enough to change practice. The key was mass production, scaling up the mold so that its antibacterial substance could be harvested consistently, purified, and supplied in usable quantities. Once production methods improved and testing showed that purified preparations worked reliably, the substance could be used beyond the laboratory. That shift, turning a fascinating lab phenomenon into a standardized, reproducible drug, addressed the very concerns that had made doctors ignore it earlier. When antibiotics became available at scale, the global impact followed. Infections that once spread with unstoppable force could be treated with targeted drugs that reduced bacterial growth inside patients. Hospitals could manage wound infections more effectively, and clinicians gained leverage over illnesses that previously depended heavily on chance and the body's own defenses. The change wasn't just about a single cure. It reshaped how medicine approached bacterial disease, transforming antibiotics into a cornerstone of treatment and prevention worldwide. How penicillin changed medicine forever. Before penicillin, many bacterial infections were treated with limited tools that rarely solved the problem. Doctors relied on antiseptics for wounds, draining abscesses, and supportive care, while infections of the throat, lungs, skin, and blood often turned fatal despite careful nursing. Even when the bacteria were known, the medicines available could be harsh, hard to tolerate, and far from dependable. Death could follow from something that sounds ordinary today, because the body's fight was often all that stood between a patient and a spreading infection. In that world of uncertainty, Alexander Fleming worked in a laboratory obsessed with simple observations: where germs grow, how they behave, and what might stop them. He prepared culture plates so bacteria could multiply in a controlled environment, and he studied the patterns with an attentive, almost patient routine. One day, after his cultures had been left undisturbed longer than usual, he noticed something strange. A patch of contamination had appeared on one plate, and the surrounding bacteria were not thriving. The mold itself looked like a quiet intruder, yet the growth around it had been interrupted as if the mold had released a chemical that told bacteria to stop. Fleming didn't treat this as a lucky accident right away, but as a clue he needed to test. He examined the boundary between the mold and the bacteria, and the contrast was unmistakable. Where the mold spread, bacterial growth faded, suggesting the mold wasn't just competing for space, it was producing an active effect. He then took material from the mold area and tried to apply it so the effect could be studied more directly. Instead of merely letting the contamination stay, he tried to capture what made the bacteria fail. At first, the scientific world was slow to care, and that made the discovery feel as if it had been trapped behind laboratory dust. The idea sounded too convenient. A mold on a plate, a barrier around it, a potential miracle in a petri dish. Yet the substance's behavior in the real world was difficult to demonstrate in a way that matched the standards of medicine. Fleming's observation was real, but developing a reliable drug from it demanded more than a promising effect on a single plate. It required consistent results, careful refinement, and methods that could be repeated by other researchers and clinicians. Without those steps, the discovery could be dismissed as a curious laboratory phenomenon rather than a medicine ready for patients. Progress later accelerated when researchers took Fleming's clue and pushed it towards something usable. The key challenge was turning the mold's effect into a stable, potent medication that could be produced reliably in large amounts. Once scientists learned how to cultivate the right conditions and purify the active material, penicillin moved from an intriguing lab observation to a drug that could be manufactured. That shift mattered because infections scale faster than individual experiments can. A hospital needs dependable supplies, consistent potency, and predictable dosing, or the best idea in the world stays only a possibility. When penicillin entered wide use, it changed medicine by turning once dangerous infections into conditions that could often be controlled. Bacterial infections that previously spread unchecked were suddenly vulnerable to treatment, and clinicians gained a powerful weapon that worked against many bacteria rather than only preventing contamination on the surface. Pneumonia, skin infections, wound infections, and other bacterial illnesses could be treated with dramatically better outcomes, not by waiting for the body to win, but by using a targeted intervention. The discovery's global impact followed from practical reality. Once production became feasible, penicillin could reach countless patients beyond the boundaries of a single lab. This transformation also reshaped how medicine thought about curing disease. The success of an antibiotic encouraged the idea that nature could hide effective weapons against microbes, and it drove the search for additional treatments. Penicillin became more than one drug. It was proof that a careful observation of how bacteria respond to a natural substance could lead to therapies powerful enough to alter survival rates. From one unnoticed mold growth on a culture plate, medicine gained the beginning of a new era, where bacterial infections no longer had to be met only with limited tools and grim uncertainty. Before antibiotics, infections were a quiet emergency that could turn into a death sentence fast. Small wounds, pneumonia, childbirth complications, even minor surgeries could spiral out of control. Hospitals had few defenses beyond cleaning, draining abscesses, and hoping the body would win. Then one ordinary mistake changed everything. In 1928, Alexander Fleming left a petri dish of bacteria unattended. When he returned, the plate looked wrong. Something had contaminated it. Not just any contamination. A mold had grown across the surface. Fleming didn't toss it away in frustration. He studied it. He noticed something remarkable. The bacteria around the mold were not growing. They were being pushed back, as if the mold had released a substance that stopped them. Step by step, his attention moved from the obvious to the proof. He observed the clear boundary between mold and living bacteria. He realized the effect wasn't random. It matched the presence of the mold. He gathered what the mold was making and tested its power against bacteria. As the results grew clearer, he connected the dots. This was not just contamination, it was a natural weapon. Fleming called the active substance penicillin, and the idea sounded almost too good to be true. But it didn't sweep the world immediately. The discovery was ignored at first, not because it lacked value, but because penicillin was difficult to isolate and keep pure, and the amounts were too small for large-scale testing. Many scientists were focused elsewhere, and the early results didn't yet have a practical recipe for mass treatment. For a time, it sat like a spark in a damp room, real, but not ready to light a fire. Then the world needed it most. During the pressure of war and the urgency to save lives, scientists worked to solve the hard parts Fleming faced, refining the production method, stabilizing the medicine, and producing enough for real patients. Eventually, penicillin could be manufactured reliably, turning a laboratory observation into a treatment that spread across continents. Doctors could finally fight infections at their source, not just manage the damage after it appeared. The impact was immediate and global. Survival rates changed. Surgeries became safer. Physicians gained time to treat, not only symptoms, but the cause. What began as an overlooked mold on a forgotten plate became one of medicine's biggest turning points, the birth of antibiotics. If you watch closely in this story, you'll see the real lesson. Progress doesn't always start with a grand plan. Sometimes it starts with noticing what everyone else considers a mistake. And as future discoveries continue to emerge from strange experiments, one question remains powerful. What valuable breakthrough is waiting right now in the data we almost ignore?